Well, thank you for that kind introduction, and it's really a pleasure to be here at the land of perpetual sunshine. When I was vectoring back to the West Coast, I sort of missed it and getting into Portland, and it's beautiful there in the summer and fall, but right now it's pretty much rainy every day, so it's great to be here. And I'm delighted to be a member of this very august list of speakers. All right, well today I'm going to talk to you about what amounts to a 16-year journey since I returned to Oregon. That was basically one of the reasons I went to Oregon was to work with people the CMV virologists there, particularly Jay Nelson, to address what I thought was a new approach to development of an HIV vaccine. And so even at that time, it was pretty obvious that conventional, at least T-cell focused vaccines weren't going to work for HIV. And it had to do with the fact that, as Rafi elegantly pointed out in his previous talk, is that with most vaccines, T-cells go down to a very, very resting state. And although they're still memory cells, they require a period of expansion, effector differentiation, and migration in order to deliver high-frequency effectors to sites of viral replication. And for many pathogens, yellow fever probably, and many others, that 2-week delay is not a problem, but for HIV it is. HIV and its counterpart in monkeys, SIV, replicates so fast that if you come in with your effector response here, it's just going to be too late because the virus is so good at evading. Once the infection is established, it's so good at mutational escape and so forth that it's almost always going to escape these responses. So, we felt that, gee, it would be nice if we could turn T cells into more like antibodies that could immediately intercept an infection right at the get-go, right at the very opening of infection, essentially have sort of a whack a virus type vaccine. The problem is, of course, that there was no vaccine that would do that. Almost all the vaccines that were being studied in this field all had this problem of too little, too late. And usually you can go to Rafi's papers and you can figure out exactly what you need to know about immunologic memory from Rafi's papers, but this is sort of the anti-Rafi Ahmed memory. We had to find a different kind of memory. And so, we couldn't return to Rafi's papers, so, I turned to another source of inspiration, which in this case is 1960s television. And those of you who are old enough to remember, there was a show in 1968 called It Takes a Thief, and the premise was that it takes a thief to catch a thief, and our premise was that it takes a persistent virus or a vector to stop a persistent virus. And of course, if we're going to use a persistent virus, we want to use the smartest persistent virus in the virome. We don't want to use one like LCMV, which that doesn't fit well in the host immune system. We want one that's been around the immune system for tens of millions of years, and for evolutionary reasons of its own, has co-evolved with its primates host to persist at just the right level to elicit robust, lifelong, effector differentiated T-cell responses in both lymphoid tissues and effector sites. And it does this on purpose. It's CMV devotes most of its life to modifying its host. It actually has genes that elicit this response. It keeps it under control, yet it doesn't clear the infection or prevent its spread, which typically is through breast milk in most places of the world. So why not cytomegalovirus? Well, why not indeed? So we looked at we knew the immunobiologic properties in human innate CMV as well as could be known in humans, where you can't do experiments very easily. We looked in rhesus CMV, which is a very closely related cousin, and we asked whether this approach made sense. And sure enough, if we started looking, we found that rhesus CMV could efficiently superinfect CMV positive animals, because most people that we were interested in vaccinating would be CMV positive, so pre-existing anti-vector immunity couldn't be an impediment, and it's not. With wild-type rhesus CMV, it basically takes one infected cell to superinfect an animal that's already CMV infected, and that one infected cell will generate an infection that will persist for the life of that animal. The other thing is that we definitely didn't want to worry about original antigenic sin, and it turns out that we could sin if we wanted to, because these vectors in the context of this superinfection would elicit and indefinitely maintain a high-frequency effector memory T-cell response to the insert, even though there's enormous CMV responses already there. And you can see here, give one vector, you give it a response, give another vector with a different insert, you get a response, and another, and so forth. And basically, we've done this about 8 times. It doesn't matter whether you give vectors together in sequence, they always work. And when they work, they elicit these very high frequency responses that are both in lymphoid tissues, but even higher levels in places like the bone marrow, spleen, and liver, and fairly high levels in the small intestinal mucosa. And this is a male animal, but we could also see it in the vaginal and cervical mucosa. These responses, unlike what Rafi showed you before, they never return back to a central memory or stem cell memory state. They stay for the life of the animal as a highly differentiated effector memory state, both phenotypically and functionally. And by the way, little to no, for whatever reason, original antigenic sin is a problem for antibodies. We don't actually see antibodies to the inserts in these vaccines, which is good from a scientific standpoint, so we don't need to worry about them. From an HIV, most of the field in HIV vaccinology is now focused on antibodies, so at the meetings I usually get put last because we're dealing with an unfavored arm of the immune response these days, the T cell response. So anyway, to test this, we decided to pick the highest bar we could, and that's the SIV MAC239 clone virus, which is like HIV on speed. It has mean viral loads and plateau phase viral loads, and about somewhere between 1 and 5 million on average, and a survival time which is about 2 logs higher than HIV, and a survival time of basically a year and a half, which is about 8-fold faster than HIV. And again, this is a T cell vaccine. We're not trying to prevent infection with it because T cells only respond to infected cells. What we're trying to do is infect animals until they're demonstrably infected, and then see what happens, and see what happens after a take of the infection. And to make a long story short, we have done this now innumerable times. My philosophy has always been go big or go home, and make sure that you're very convinced of your effect. And here is our circa about end of 2015 sum total of Effector Challenges, and you can see the controls have the typical, and there's a bunch of them here, 67, have the typical kind of peak viremia, then a very high plateau phase viral load. One animal got infected sort of occultly and then spontaneously burst off. But for the most part, they all look very characteristic. This is an Ad5 vaccine, which shows the fact that you get the same infection, only it's a little bit lower plateau phase viral load. And this vaccine is no longer in favor anymore. In contrast with CMV vectors, about 54% of the animals combined, 164 out of 119, have a very different effect. They have a plasma viremic blip, typically, and then they go down to complete control. Sometimes they blip afterwards, but for the most part, the virus, they're aviremic during the vast majority of times in this state. We've had one animal recrudesce here out of a total of 65 that were originally protected in these studies, but still overall 6-month efficacy is 54% of animals with this complete virologic control versus 0 of 130 controls, which is a 2.2 times 10 to the minus 16th p-value. So, people are a little bit nervous about giving wild-type CMV potentially to humans, so we've also worked on an attenuated vector. The one that's our favored one at the moment is one that's missing the PP71 gene, which results in a 2-log growth deficit and multi-step growth curves. Little to no vector dissemination from the site of inoculation, no vector shedding and secretions, and no vector transmission with co-housing or transfusion. We've tested this in both a heterologous and a homologous challenge, side-by-side comparison, using either MAC239 inserts or E660 inserts, or about 15% amino acid differences across the genome. The immunogenicity of the attenuated vector, if you compare the blue lines, light blue and dark blue, is about the same. The heterologous one's a little bit less because the peptides don't match. But nevertheless, when we challenge animals, we see the same protection with these attenuated vectors. 54 is actually a little bit higher with the heterologous inserts, but that's not significant. But sure, they're about the same. So let's take a little closer look at what's happening, because this is unprecedented protection against a very highly pathogenic AIDS-causing lentivirus. And if you look more closely at the protected animals during the acute phase, you can see that there's actually often detectable viremia over a 2 to 4 day period, starting at about day 7, day 8. And then going away, and then at this point they're completely aviremic, even with a very high level, less than 1 copy detection limit. And you can see that, and this probably reflects a virus that's made in the tissues about 2 days before. So basically the infection is stopped somewhere between day 4 and day 8 in all these protected animals. But if you take animals to necropsy at this stage or even a few weeks later, you can easily find virus. The red is, the red is RNA, the blue is DNA. It's not a lot of infection, but there's definitely infection. It's in the site of inoculation. This is the rectum colon. It's in the draining lymph nodes, mesenteric and ileosacral, and it's in the bone marrow, spleen, and liver. And that's pretty consistent. We can find live virus. We can co-culture virus out of these tissues as well. It's not nearly as high as progressive infection, but these protected monkeys are definitely infected in the tissues. And in fact, as shown here, if you take in this case bone marrow cells, and I can't remember, it's like 10 or 20 million bone marrow cells, and you put them in a new animal, a naive animal, they will transfer the infection at this point, and we've even shown adoptive transfer of infection through day 56 after the onset of protection. Okay, so let's take a little closer look at this. Again, the fact that we don't put SIV VIF into our vaccine vectors. So when you see an SIV VIF response, that reflects exposure of the immune system to the actual SIV infection. And in fact, all the animals, both protected and non-protected, develop very strong, at least early on, SIV VIF responses. Again, showing that there's an ongoing infection that's controlled. If you look at one of the things that happens with HIV and SIV infection is systemic immune activation. It usually starts somewhere right after the first week of infection. This protection, we use CD169 induction on circulating monocytes as sort of a poor man's way of monitoring immune activation. It's actually quite sensitive. And in fact, you can see the control. The unprotected animals show upregulation in immune activation, whereas the protected animals don't. So the protection is before systemic immune activation. And the other thing in keeping with our original hypothesis is that there is no anamnestic response with these effector differentiated responses. The responses to the vaccine inserts stay completely flat after the onset of protection. And so clearly whatever protection is occurring with responses that were pre-existing and maintained by the vector at the time of challenge. Just in case anybody was wondering whether we might have antibodies to OMV that we couldn't measure that we're still protecting, we can easily, all those previous studies had OMV inserts as well as GAG, Rev, Nev, TAT, and POL inserts. Here we just have inserts to POL, Rev, Nev, TAT, and GAG, and again we see the same level of protection, and we can even give the vector orally, and it works just as well. And again, this will come back to this in great detail later. There's no correlation of this protection with MHC1A genotype. Okay, so now the thing that we weren't expecting, we were hoping for everything I've showed you so far, because that was the hypothesis that we could, an effector differentiated response could provide protection because it's on board at the beachhead, so to speak. But this is what we weren't expecting, and that is if you follow these animals, you can see here these are actually the combined groups that I showed you before, that the Delta PP71 vector. You can see that the VIF response goes up, and then it starts going down. And by a year to a year and a half after the onset of protection, it goes away till it's no longer detectable. And it's no longer detectable in tissues either, let alone just in the blood. If you follow virus, and we use the bone marrow as a convenient place where it always is early on, and then we follow it over time, it's easy to get to bone marrow. And in fact, we'll see positive, we'll be able to isolate virus fairly readily from just about every animal at 4 weeks, at 8 weeks, 12 weeks it starts going down. At 20 weeks, we don't see it anymore. And if we continue following these animals, we don't see virus in any of the places that we look. In fact, if you take animals to necropsy, between 1 and in this case 3 years after challenge, you can see that basically you can't tell them apart by a very extensive quantitative nested PCR analysis. You can't tell them apart from an animal that has never ever been infected. Very different from an SIV positive monkey on ART. Basically, we can't find virus here, and we cap that off by actually doing adoptive transfer with 60 million cells from these animals, and show that while controls on ART or elite control is easily adoptively transfer adoptively transfer infection. And as I showed you, early protected animals also easily transfer infection. These late protected animals do not. So basically, the virus essentially disappears. And this is the first sort of immune-mediated elimination of AIDS-causing lentivirus. And again, these viruses, one might argue whether the virus is really eradicated here, but certainly these infections are functionally cured. In fact, so much, we've gone back and re-challenged animals that had been previously cured. And here's a group of 8 female animals that were re-challenged intravaginally. They all got infected again, but I see the VIF response going up. Interestingly, only a few of them showed some blips, but 7 of these 8 were protected, were able to control and ultimately eliminate the second infection. And another 6 animals with intrarectal were cured from intrarectal challenge, were re-intrarectal challenge, and 3 of the 6 were protected, again, as shown here, including one that was almost 7 years out from its original vaccination. So the conclusion is unlike many other vaccines that people use in monkeys for HIV, these vectors, the immune response and protection is exceedingly durable. Okay, so let's just summarize where we are because we're not even to the good part yet. These recent CMV vectors have a unique pattern of protection, what I call control and clear. It's binary. It's either complete or none at all. Again, it sort of reminds one of what would happen if you're defending the beachhead and you're not reinforced. If the enemy either stop the enemy in the water or the enemy gets behind you, you lose. And that's the way this protection seems to be. It works for intrarectal, intravaginal, and even low-dose intravenous challenges. So it doesn't seem to require a mucosal challenge. It's extraordinarily durable, as I mentioned. The efficacy is maintained with a pretty profound vector spread deficiency, and antibodies or OMV inserts not required. And as I told you, and I'll come back to it, it's independent of MHC1A genotype. And that leads us to a very important question, if we're going to translate this to humans, is what exactly is the mechanism of virus control and clearance? And from very practical questions for development, what determines protection versus non-protection? Is it stochastic chance? Is it monkey genetics? Or is it hopefully differential vector immunogenicity, which is something that we could modify and perhaps increase efficacy? And that's the question. Can we increase efficacy by understanding what the mechanism of protection is and optimizing vector design? I mean, 54% is good, especially against a very highly pathogenic SIV, but we'd always like to do better. Now, some of the problem with doing this is that when we first looked at just overall measurement of SIV specific responses, either at the peak or pre-challenge, and this is, I think, like 7 studies where we've done immune correlation, you can see this forest plot that basically the outcome does not seem to correlate with either of these responses in blood, either peak or pre-challenge, and we've also done it in lymph node and other accessible sites and haven't seen a correlation either. So that suggests, the overall response isn't correlating. We think this is T-cell mediated because there's no antibodies. And so we wanted to look into this a little bit more closely. And in fact, about the same time, we came across something that was quite spectacularly different about these vectors than what has ever been shown before with any other infection or let alone vaccine. And that is, it's basically going down into the immunologic wonderland here. And some of this was published in 2013, and another recent paper basically we've shown that the epitopes that are targeted by the CD8 T cells elicited by these vectors, these strain 68.1 recessive vectors, are entirely non-overlapping with conventional epitopes that are elicited by SIV itself or by conventional vaccines. And so basically, what everybody in this room knows, I'm sure, is that when you get a natural or a conventional CD8 T cell response to conventional stimuli, and you look at the epitope level, what you'll see is, and these are peptides, overlapping peptides to SIV GAG, 11 amino acid overlap starting at 1 and going to like 125. And we just test each peptide and we determine the restriction either by blocking or by transfectants. This is mostly by blocking. And we show, not surprisingly, that these responses, these epitope responses, are all MHC1A restricted, except for there's actually one here, right here. You can occasionally find an MHC2 restricted response. It has been shown in humans as well by Bruce Walker's group. And if you look at the distribution in the population level of these responses to these various peptides, you can see that with the exception of this one spike here, which is actually due to the fact that a particular monkey MHC1 allele was overrepresented in this cohort, you see that they're really diffusely distributed with no response in these outbred monkeys, much over, most of them are 5 or 10% and occasionally 20%. Most of these are also subdominant MAMA1 responses. So, this is exactly what you'd expect. This is not what we see with 6-strain 68.1 recent CMV vector elicited responses. And you can look at it, this is going to be very bright. And this is what we see with these responses. This is a number of animals, I don't know how many, 30, 40, whatever. And you can see that none of these responses are actually restricted by MHC1A. They are entirely restricted by either MHC2, just like a CD4 T cell would be, or by the MHC1B molecule, MHCE. This restriction was determined by both specific blockade with its specific inhibitors like the VL9 peptide or the CLIP peptide, as well as by validation on single molecule expressing transfected APC. And you can see here that some interesting patterns. Not only are these responses extraordinarily broad, these are actually the epitopes. These are the peptides, but remember some of these peptides are like these two here are probably one epitope. The independent epitopes, there's at least 50 combined epitopes in the GAG molecule on average in these responses. And if you look at some of them, you see here there's like lines that go all the way across all these monkeys. And you can see it here easily, and I point them out to you. Each one of these boxes is present in all the monkeys, and these are so-called supertopes. They're present in 100% of outbred monkeys. And you could look at it in a different way, looking at the frequency plots. You can see that here are the supertopes. We actually determined the optimal 9-mer for these supertopes and found out in 130 of 130 monkeys, we could detect that were vaccinated with this vector, we could detect these responses. So these are uniform, invariant responses that are always present in these animals. And again, so this is an entirely new kind of immune response. There's frequent supertopes, both class MHCE restricted and MHC2 restricted. And even the ones that aren't supertopes tend to be cluster and tend to be at higher frequency, 40% rather than 5 to 20% across the board. So this is an entirely new kind of CD8 T cell response that has never before been reported. We went back in with, as I mentioned, with single molecule transfectants, looking at monkey MHC2 molecules and found, again, confirming that these were actually MHC2 restricted and showed that the supertopes generally resulted from promiscuity of the peptide epitope to multiple MHC2 allomorphs. And you can see here just an example of one peptide which is presented by 3 different allomorphs. So basically every monkey had at least one allomorph that could present the supertopes. For MHCE, it's even more interesting. We'll come into the biology of MHCE. But basically, MHCE, which is relatively non-polymorphic, could present all of the MHCE epitopes. The same molecule could present all the epitopes, and even HLA-E could present the same molecule. So these relatively non-polymorphic molecules were able to bind diverse peptides, and we'll come back to that in a little more detail, and present them to the T cell. And just to show you that this isn't a peptide artifact, we actually can measure the response to SIV infected cells, to both purified CD8 T cells. These are alpha beta T cells that are purified. We put them with infected SIV infected cells, and we either block them with no block, or we block them with this VL9, which is anti-MHCE, or CLIP peptide anti-MHC2. The SI-MAC239 infected the T cells from those infected animals are not blocked by VL9 or CLIP, or MVA GAG not blocked by VL9 and CLIP, but these responses are entirely blocked by VL9 and CLIP in the rhesus CMV strain 68.1 vaccinated animals. Okay, so what is MHCE? This is a fairly sophisticated audience. You've probably heard of it, but it's a class 1B molecule, which is the most conserved MHC1 molecule in primates. And I'm losing my arrow here. It's the most conferred MHC1 molecule in primates. Its primary job is to present a 9-mer peptide. The canonical peptide is called VL9, a very high affinity peptide for MHCE, which is derived from the leader sequence of MHC1A molecules. It presents them to inhibitory CD94 NKG2A receptors on NK cells, and basically it allows NK cells to determine whether a virus has infected that cell and downregulated MHC1. Because if you downregulated MHC1, then you don't get the signal anymore, and the NK cells lose the inhibitory signal. This effect seems to be sufficiently important that smart viruses, including both HIV and CMV, upregulate MHCE while they're downregulating MHC1. So you could, you can see this here. MHC is upregulated in SIV infected cells while MHC1 is downregulated. And this is entirely to counter the NK cell response. MHCE can restrict T cells. They have been reported to restrict T cells here and there, relatively rare for most responses. And they typically involve recognition of peptides, which are very similar to the canonical VL9-like epitopes. And so certainly not the breadth of what we see in these vaccinated animals. And by the way, these T cells are not recognizing MHCE by NKG2A, as we can see that the T cells that are doing the recognition of these peptides are CD3 positive, CD8 alpha beta positive, TCR gamma delta negative, and the vast majority of them are NKG2A and C negative. So this is sort of interesting. What we found is that MHCE, this relatively non-polymorphic molecule, which has been implicated in all sorts of things, but certainly not responses to most pathogens, we see this extraordinarily broad response. On average, about 20 MHCE epitopes in these vaccinated animals, and that's compared to 11 in unconventional vaccination and 14.5 in controlled SIV MAC239 infection. And these are restricted by all of the up to like 20 or 25 MHC1 molecules that monkeys can express. So, and this is really, there's only two or three loci for, probably only 2 loci for MHCE, and they all present the same peptides. Again, if you look at this pattern, you can see that of the 125 peptides that are in GAG, 15 peptides that are in GAG, about how many of them? 109 of them have at least one MHCE restricted response within them. So it's very, very broad. And in fact, these are the optimals. These, again, showing these are T cells. These T cells can detect either on BLCL or on transfectants. They can detect these peptides at less than 1 nanomolar concentration. So they're very highly avid T cells. And yet none of these optimal 9-mers match the, well, I shouldn't say none of them. Only one of them matches the canonical sort of anchor residues that has been described for MHCE. The rest of them are very diverse. In fact, there is no there is no favored amino acid sequence. They're basically binding peptides across all classes of peptides in all of the amino acid positions. And we have indeed shown that all these peptides, well, actually all but one, were demonstrated in molecular binding studies to bind HLA-E. And we turned to Dr. Nanakaran at Los Alamos Labs, who's a modeler, and he basically showed that all these peptides actually do fit inside and bind to, in document analysis, to both HLA-E and MHCE based on published structure studies of HLA-E. They all can bind. Theoretically, and they all have the same backbone conformation when they bind. He also shows something very interesting about HLA-E, which wasn't previously appreciated. HLA-A, conventional MHC class 1, collapse when you remove the peptide, where HLA-E is a very rigid structure that doesn't collapse, allowing that this peptide binding groove remains open, allowing for potential peptide exchange. That will become important later. And then what he's also shown is that the epitopic peptides actually sit higher in the groove than the canonical VL9 peptide, are more solvent exposed, and actually encounter a very diverse binding environment in terms of charge, polarity, and so forth. And he's calculated that the contribution to binding by the various amino acids in these peptides can vary. Sometimes a polar amino acid does it, sometimes a basic, sometimes acidic, sometimes not. So it's a very varied binding environment. And there's more details of this in that recent 2016 paper I referred to before. But the bottom line is, from an immunologic standpoint, is MHCE can bind and present an enormously diverse pool of epitopic peptides. In fact, in all these different, very unrelated TB and SIV antigens and even rhesus CMV antigens themselves, about 4 distinct epitopes per 100 amino acids on average can be restricted by MHCE and presented to T cells when the animal is vaccinated with these rhesus CMV vaccines. So how does this happen? This is really weird. So, how does CMV do this? We've already acknowledged it's the smartest virus in the room, but this would warrant professor status even at Emory, I think, if Rafi's still here. And the bottom line of this is that wild-type CMV doesn't do it, which is good. I have to say, when I found this out, I was happy because I had been studying HCMV and wild-type CMV for many years, and I've never seen a hint of this. They've all seemed to be conventional MHC1A restricted, and in fact, wild-type rhesus CMV is, the responses to IE, as shown here, are conventionally restricted. However, if you take the vector and give it to a naive animal, a CMV negative animal, you see that the IE responses look just like those gag responses I showed you, entirely unconventional. And if you take the vector and give it to an animal that already has these responses, you basically see a combination, just as you would expect. So clearly, there's something different about our vector that creates these responses. And now this vector was when we started working with it 16 years ago, was the only one that was available. It was a highly fibroblast adapted rhesus CMV that we had a back for to make the vectors. And it's missing, as are most fibroblast fibroblast adapted CMVs, it's missing parts of the pentameric complex. In this case, the monkey rhesus CMV equivalents of UL128 and UL130. During fibroblast culture, this pentameric complex, which is a receptor for non-fibroblasts, basically slows the virus down. Let's go back here. Basically slows the virus down, and it's usually lost by deletion, but a variety of ways the vector can lose this. And sure enough, this one, our vector is missing this component, this 128, 130. And if we go on to if we take the strain that we started with, 68.1 and repair expression of these two molecules, sure enough, we converted entirely to classical restriction. And again, no supertopes, nice responses, all MHC1A restricted. And if we are good virologists and then go back in and redelete these molecules just to make sure it's specific, sure enough, when we redelete these molecules, the weirdness comes back, and supertopes and the MHCE and class 2 restriction comes back. So this is interesting because this allows us to ask the question whether the CD8 T cell unconventional epitope response matters. I mean, is this just a sideshow? And an interesting sideshow, but it could just be a sideshow, right? So let's think about what the potential advantages of this unconventional response would be, especially the MHCE responses. But certainly we have very magnified or expanded response breadth, unprecedented response breadth. We have recognition promiscuity, so this would allow consistent epitope targeting in all or most vaccinees, unlike typical T-cell responses where you have to worry about whether they're A2 positive or whatever. It would allow for tolerance of mismatch between vaccine insert and field strain sequences because there's always going to be a match somewhere. This non-overlap with conventional responses would be potentially advantageous in settings involving escape variants because these are not natural responses, at least not typical responses. The virus will not have escaped them prior to vaccination, either in a therapeutic setting or even with transmission of escape variants from person to person. So we'd have that advantage. The other thing I've already mentioned is MHCE is expressed at very low levels in normal cells. In fact, you could barely see it or not even see it at all by immunofluorescence, even though it's expressed. But it's really upregulated, brightly upregulated in infected cells, sort of standing out as a beacon. Here I am in an infected cell. MHC2 class 2 restriction would facilitate targeting of infected MHC2 expressing targets like macrophages and DC and some activated T cells, and would also, for better or worse, provide for T cell triggering by uninfected professional APC, which might be an advantage more for TB, as I'll come back to, than for HIV. So anyway, let's get to the answering the question. So the question is does the UL128, UL130 repaired strain vectors protect compared to our original vector, which we know protects? It's a fair comparison since the immunogenicity of these two vectors is otherwise the same in terms of magnitude. It's otherwise the same in terms of phenotype. They're both, this is 68.1.2, this is 68.1. This is the percentage that are effector memory, transitional, or central memory, and you can see they're exactly the same. Even the distribution in tissues, looks to be, close enough for government work. So the only thing that's different that we can tell about these vectors is really the restricting elements of the CD8 T cell response. And so we asked the question whether they protect or not, and in fact they do not. So the conventional responses, the conventional MHC1A restricted responses show no protection at all in 2 separate experiments. And again, as always, the 68.1 vectors show the classic sort of 50 to 60% protection. So this is very interesting. It creates some translation problems, but it's very interesting. Either the MHCE or the MHC2 restricted CTs are a critical component of the protective mechanism. Okay, so can we determine which is the right one? Personally, I have some data to suggest this. I think it's the MHCE responses, but it would be nice to be able to show definitively one way or the other which of these are protecting. And in fact, I think we can do this because the basic question is can we separate these responses in different vectors, and are these responses independently regulated? Well, this is actually an area which I don't have time to go into great detail, but I can tell you that they are independently regulated, and I'll show you one example of that. So I mentioned to you before that CMV upregulates MHC in infected cells, and it does that to escape NK cells, and it does that by expression of a rhesus 67 or UL40 ortholog, which encodes a VL9 dimer and actually serves, once the US2 through 11 genes downregulate MHC1A, it serves to provide the VL9 peptide to MHC and allows it to be not only expressed but super expressed. And in fact, if you knock out rhesus 60, this molecule from rhesus CMV, you no longer upregulate MHC in infected cells, as shown by this dark line compared to this line. One of the things I'll point out to you here is that when you look at HLA-E, actually this is not HLA-E, this is MAMO-E, but nevertheless, you look at its expression in an infected cell, you can see a lot of the expression. You don't see that much surface expression, although it's definitely there, shown here. You see it in this Perry-Golgi apparatus, which actually is the viral assembly complex, and you see it right here in vivo. High upregulation of MHCE in these infected cells in vivo, and in fact that's where we think peptide exchange occurs that allows the epitopic peptides to be loaded onto MHCE. But nevertheless, without this molecule, this doesn't work. We asked the question, what happens to the responses if you knock out the rhesus 67 molecule? And lo and behold, just as you'd predict, deletion of rhesus 67 from the vector is interesting. The vector it has so many immune evasion molecules that it still infects and is still immunogenic, just like normal, as shown here. But now the responses are entirely MHC2 restricted. We've lost the E responses, we've lost the E supertopes. And so now we have a vector that, strangely enough, makes class 2 CD8 responses. Very strange indeed. Now, this is just the tip of the iceberg. We've already made vectors with different gene manipulations that make various percentages of conventional versus unconventional responses. We can differentiate between so-called canonical MHC1A responses and non-canonical responses with US11 deletion, which is something that's in that first 2013 paper. And we've made a variety of, vectors that can be MHC2 with supertopes or MHC2 without supertopes by manipulating tropism. And we have a high priority as vectors in MHC only vector, which is in progress. We actually have a number of strategies. We think this will result in an MHCE only vector. So our plan is to construct, validate, and efficacy assess rhesus CMV vectors with all the different flavors of T cell responses. And this is, funder willing, because these are expensive studies. And the goal is to develop an HCMV HIV vector that can elicit the most efficacious response flavor in humans. And that's our next big challenge. We are, in fact, in GMP manufacture right now of a HCMV HIV ortholog of the rhesus CMV 68.1 vector. So this is delta PP71 HCMV HIV gag vector, which is missing the UL128, UL130. We hope to be in clinical trials in early 2017. Manufacture willing, because again, that's always a hard part about doing human studies, is manufacturing at GMP level. And we will see what happens and report back on that. However, HIV is not our only interest with this vector platform. The most advanced other disease is tuberculosis. We have now completed one study and are almost complete with a second study, which shows actually quite remarkable control of tuberculosis. Rhesus monkeys are very susceptible to tuberculosis. It's basically 100% fatal in this species, as opposed to cyno CMVs or humans, so it's a very aggressive disease. And in the first study, with a strain 68.1 Rhesus CMV TB vector, we got 73% efficacy against disseminated spread, different statistically from both unvaccinated and from BCG vaccinated. And BCG actually abrogated or partially reversed protection, so BCG is not completely good for you. We've now repeated this study, and we repeated it both with 2 different 68.1 vectors and a 168.1.2 vector. And now we're seeing animals that are clearly have been infected with TB but have no disease at all. They're completely, have no lung disease, and we're taking these necropsy. In fact, they have no disease at all, even though they develop nice T-cell responses to many TB antigens. They clearly were infected. So this is, again, it looks like both, in this case, both 68.1 and 68.1.2 protect. And in fact, the primary correlate in this study was a CD4 T-cell correlate. So we're also interested in therapeutic HIV cure. We have some data coming out in the next few months that'll see whether that works there. We have one experiment in malaria showing some protection at the liver phase. We're working on thinking about hepatitis B, hepatitis C, and herpes simplex. And finally, obviously, MHCE is upregulated in a number of cancers, and so we're thinking about taking this and looking at their efficacy in the cancer world as well. So with that, I'm going to stop. I need to thank some key people. This is I have a very large group of people that contribute to this work. All the monkey work in immunology and efficacy studies were managed by Scott Hansen, who's now an associate professor, but still works in our group to manage our large monkey studies. Klaus Frew and Jay Nelson are my virologists. Particularly, Klaus Frew makes almost all the vectors that we use. Jonah Sasha is a young faculty member who helped with the MHCE story. All of my SIV work is done in collaboration and partnership with Jeff Lifson, and based on assays that were developed by the late Mike Piatek. And finally, a number of other people have contributed to like the MHCE story and Andrew McMichael's group and so forth. And I have to thank my parents, my funding parents. Like most, I've never grown out of having my parents fund me. So on the NIH side is Michael Pensiero and Alan Schultz, and on the Gates Foundation side, Pervin Ankelet Cesaria and Linda Stewart, keep me in gas money. And we do have a small startup company that is trying to develop these vectors for clinical translation. And with that, I'll stop and take any questions.
Hi, Louis. That was nice. I have actually 2 related questions. The I'm confused about how the UL40 works. That I thought provided a peptide that binds to HLA, right? So, yeah, would that not block the groove?
So UL40 has an embedded VL9 peptide, which is essential for its use. If you knock out the ability to bind MHCE, but what it does is it upregulates MHCE. It takes it to that compartment. We think it takes it to that viral assembly compartment, and then the peptides exchanged. The epitopic peptides exchanged in that compartment. They're very high, really high CMV proteins in that compartment, and then it's taken to the cell surface with the virus, actually.
Gotcha. So maybe that provides answer to my other question, which is, would you expect to be getting responses against self-antigens in this environment where you're getting such promiscuous?
We don't think these responses are the self-antigens. These are all completely SIV specific, and we haven't seen evidence of cross-reactivity, but we can put self-antigens in these vectors and get rid of these responses.
So is that because the self-antigens are not in this viral assembly compartment, do you think, or some other reason?
Well, no, so we can take vectors that express self-antigens. No,
I understand, but when you don't have the vectors, it's because the antigens are not in the virus.
Well, yeah, I mean, you don't get these responses to this degree in anything other than CMV, right? So, CMV is bypassing whatever the restrictions are that prevent these responses normally from occurring. It's bypassing those and allowing us to make responses to virtually anything. Self-antigens, cancer antigens that have these characteristics. What I think is the canonical MHC1A epitopes are tolerized, but these MHCE responses and class 2 responses and even non-canonical MHC1A responses don't seem to be, and we can make the same exact responses to self-antigens. Now, whether those responses do anything, one thing, MHCE is very low on normal cells. it's upregulated inflammation, it's upregulated with infection, but it's very low normal cells. And so, we haven't seen any evidence that any clear evidence that those self-antigen T cells are doing anything. But we're just at the very early stages of looking at that. Over here.
Great observations. I just had a question. If you could speak a little bit more about the safety of using CMV as a vector in regards to, CMV potentially causing other illnesses in humans, and what exactly the Delta PP71 is doing.
Well, PP71 is a viral inhibitor of a host inhibitor called DAX. Okay, I didn't have time to go into it. So DAX is an anti-herpes virus thing, one of these intrinsic inhibitors of herpes viruses that binds to IE1 and it stops basically viral growth. Okay, if you have DAX will force the vector basically to go into sort of a latency type, which is not a complete. Which doesn't mean it doesn't make any genes. It just doesn't. It doesn't have a lytic infection, so it prevents lytic infection. And so when we put and you could overcome that at very high MOIs and get the first round to work, but then the virus that makes isn't very infectious because it can't start. Low MOIs, it can't start a new infection. It can go in there and express and spread a little bit and express the insert, but it can't. But it really doesn't spread. And in fact, I didn't show the data, but if you put it into a monkey, it basically stays at the site of inoculation of the draining lymph node. You don't really see any disseminated vector. So it's quite, and it's not, you could take blood out from those animals and put them in another animal. The wild type will transfer, but the PP71 won't. We're also the UL128, UL130 deletion is a tropism receptor that also is attenuating and probably prevents the vector from crossing the placenta, which would require epithelial infection. And finally, we're also introducing microRNA restrictions on these vectors that prevent them from going to neurons to help as yet a third layer of protection against it when we put them in humans. Remember, initially we're going to go into, most of the time we're going to be going into people who already have the wild type vector. And in fact, the human vector is about 4 logs attenuated compared to the wild type vector. So I'm actually not so worried about safety. I'm more worried about being able to manufacture these things at scale and more worried about whether we can recapitulate this unusual immune response in people. Yeah.
Yes, the HLA-E restriction of CD8 T cells is so extraordinary. Are you able to block that with antibodies to alpha beta T cell receptor, or do you know it's the T cell receptor?
We're actually cloning the T cell receptors as we speak. It's a little bit harder in the monkey because nobody has developed those techniques, so we're actually doing that. I just got an email yesterday that we have our first alpha beta T cell receptors, but I haven't seen what they are yet.
So one of the things
we can block it with the VL9 peptide. That's basically what we use for the specificity control.
Right, but I think David showed 20 years ago that really all MHC molecules can restrict sort of all T cell receptors at some low avidity. Do you think it could be that actually HLA-E is going on the surface and it's stabilized on the surface by UL40, and then that serves a co-stimulatory or adhesive role that enables otherwise subclinical or very low affinity interactions with T cell receptor to be recognized?
Well, except for these aren't low affinity interactions. As I told you, these T cells can recognize sub-nanomolar amounts of peptides. So
yes, but you don't know the T cell receptor is doing it.
We don't know which T cell receptor is doing it yet. We will know that.
Or that it's even blockable by antibody to alpha beta T cell receptor.
Well, in the monkey, there are no antibodies to do that with, so I can't answer that question. But that's the bottom line. Is I don't know how CMV breaks this tolerance. It just at this point it just does. And these responses recognize. Remember, they recognize infected cells, SIV infected cells. One more over here.
Hi, right here.
Yeah.
Are the responses limited to pathogens that actually upregulate MHCE, or do you think you can?
No, I mean the responses that we elicit with CMV are to anything we put in there. The kitchen sink will make these responses. Okay,
but
whether they're going to work, they're going to work or not might be related, to that. To that. I mean, but fortunately, a lot of pathogens that we're interested in, hepatitis C, have been shown to upregulate. HIV have been shown to upregulate MHCE. So in effect, we're taking advantage of a pathogen's adaptation, immune evasion strategies with these E-restricted T cells. So they evolved to upregulate E in a situation where they didn't have to worry much about E-restricted T cells, but now we're going to give them a boatload of E-restricted T cells to deal with. And we're hoping that allows us an edge against the pathogen, which otherwise vaccines haven't worked for.
Mila.
Yeah.
These cells in which you have these CMV vectors replicating, to what extent is the antigen presentation pathway affected? So in other words, is TAP normal? Is the assembly of peptides occurring?
You mean in infected cells?
Yeah.
CMV infected cells?
Well, in your vector, with this particular vector.
Yeah, it's CMV is completely changes everything about antigen, in infected cells about antigen processing. You know, it markedly upregulates class 1. It has all sorts of regulators of processing that are intrinsic in the virus. So it's really a master at disrupting that, and that's why the net effect is what we're seeing. Now, have we dissected that yet? We haven't yet. We're in the process of doing that.
What's the function of the class 2 restricted CD8 cells?
I don't know what all I know is that they're there. I don't know if they're doing anything yet. I suspect that it's the MHCE responses that are the ones that are protective, but I don't know that for sure. But I will know that in about another year because I have these vectors that only make class 2 responses. If they're like dishwater or PBS, as Rafi says, and they don't protect, then I'll know it's the E responses. And I'm hoping we'll have an E vector that does only E responses within the next few months, in which case I'll know very shortly after that whether it is the E responses and whether focusing the responses on E actually increases efficacy, which is the goal.
Okay, last question, Rafi.
Louis, you've probably shown this before, and I may have missed it, but have you formally shown that CD8 T cells are doing the protection? Like, have you depleted CD8 T cells prior to challenge?
No, that's a difficult thing to do because these challenges take weeks and weeks. We actually now have a resynthesized antibody. We're about ready to start that experiment. The only evidence to suggest that it's CD8 T cells is really this, the fact that you switch the epitope and you lose protection. But we do have, if you CD8 deplete after they're protected, you don't lose anything because the infection is so suppressed that before, yeah. We have to maintain the depletion for like 2 to 3 months, and we've only now been able to do that.
Okay, great.
Thank you.
Let's thank Louis for a great talk.